Good morning and welcome to Mission Bio's virtual webinar series. We're so happy that you could join us this morning for this wonderful introduction on the Tapestri platform given by our own Gemma Fuerte, who is the European Field Application Scientist. We'd love to answer your questions in real time, and so please use the chat window or the Q&A box to ask any questions, and we'll get to them during the talk. And just as a quick reminder, we are also recording live on YouTube. So Gemma, how are you doing?
Very good, thank you. How are you?
Good, good. We're excited to hear you speak today.
Awesome, thank you so much. Thank you everyone for joining today. So as Kelly said, we will talk about exciting new things and also an overview of the Tapestri platform. So let me just get started. So what is the agenda for today? So we will have a background of why single cell, and also we'll do an overview of the Tapestri platform. After that, I will show you a brand new publication, so about therapeutic resistance to a drug, and then we move on to multiomics. So I'll show you SNV plus CNV and SNV plus CNV plus protein. So for those who know me, I know there is a lot of people I know already. I always like to start with these slides because we know that cancer is very complex, and we know that within a cancer mass, there are probably many different populations that might have different advantages based on perhaps somatic mutations only, or somatic mutations and also some protein characteristics that make them special, right? So at Mission Bio, we are so committed to provide the exact clonal composition of a cancer sample. So if we think of this ecosystem, of this cancer ecosystem, we see there are different populations here based on that contain different somatic mutations, so carry different somatic mutations, and some of them co-occur, right? This is something that you could only see with single cell, and that's what I will show you with real data. Not only DNA in terms of somatic mutations, but also copy number and protein expression markers. So how do we achieve this? So of course, the traditional way to analyze DNA is bulk NGS, or population-based analysis. So from the bulk NGS, we have one sample, we have millions of reads, and then we provide you what is called a VLEL frequency average of the mutations. So we tell you that percentage is mutant for IDH1, or that percentage is mutant for IDH2, if we follow the example on the screen. With the Tapestri, we barcode each and every single cell prior to sequencing in a way that then we can tell you exactly which mutations are present in which cells, thanks to individual barcodes, unique barcodes. So if we follow this example, we see that, and this is exactly the same sample, right? We see that IDH1 and IDH2 are actually present in majority of the cells below. And that's why we have no problems to detect these mutations when we use bulk sequencing, because for populations that are present in a large percentage of the cancer sample, there is not usually a big problem. But the problem comes when the mutations are present in a very small percentage of the population. We are talking about 1%, 0.1%, such as NPM1 here, or TPS3. So when the mutations happen in very small percentage of the population, bulk usually miss that. So we are able to detect them with a single cell technology. Moreover, we provide you the zygosity. So we tell you if the mutation is homozygous or if it is heterozygous, something that bulk NGS cannot do. If bulk NGS is telling you there is a 50% allele frequency for one mutation, you don't know if 100% of the cells are heterozygous or 50% are homozygous, right? So that's something that with single cell, we can provide you with no problem, and then you don't need to infer your data anymore. So I'm very excited to show this figure from a recent paper from Nick Navins, Dr. Nick Navins and MD Anderson. This is a review, and they talk about how DNA, RNA, single cell DNA, single cell RNA, and epigenetics can be used in cancer research. So in terms of DNA especially, he reviews this technique, and he uses for these different highlights that you have here on the screen for their own cancer research. So of course, single cell DNA can help you resolve the clonality within each sample. That is what we have here, the clonal structure. You could also draw phylogenetic trees, You can follow the tumor evolution during treatment and clinical stages, so see how clones change at different time points. And of course, the mutation co-occurrence and zygosity. That's what I was saying before. So we tell you whether the mutation is heterozygous and homozygous and whether they co-occur in the same cell or a different cell, which has extreme importance in drug resistance, as we will see later in the example. So in terms of more specific details about the Tapestri platform itself, so we are the only single cell platform that provides SNV, copy number, and protein at a scale from the very same cell. In terms of how many loci you can target, it depends. So we can detect SNVs and indels across hundreds of targeted loci. The minimum number of loci is 20, so you can generate a panel as small as 20 amplicons or as large as 1,000 amplicons if you wanted to. What is the cell throughput? So the cell throughput is around 5,000 to 10,000 for DNA only. And as you add analytes such as protein, we see anywhere between 2,000 to 10,000 cells. That's the output. And you might be wondering, what is the input? So the input, the recommended input is 100,000 cells. So from 100,000 cells, you get 5 to 10,000 cells out. What is the sensitivity? So we have high sensitivity for rare clones down to 0.1%. So if 0.1% of your population is different, we will be able to detect it. Even more, so we can be even more sensitive when working with different time points. So we have seen real data, published data, that when we work with different time points, you could go even lower to 0.05% detection of like a clinical remission sample in an MRD scenario, for instance. What about the content? So our panels we have some catalog panels that I will show you later, the examples, and you could also create your own. So we are very flexible. We work with human and also now with mouse. So you can create a panel for mouse if that is your area of interest, as well as human. And then I'll show you a tool, a very friendly tool that we have to create your own custom panel. In terms of sample input, we can work with whole cells, of course, cells in suspension, and we could also work with isolated nuclei, which is a huge advantage over other single cell techniques such as RNA sequencing, because you can work with frozen tissue and just extract the nuclei and work with the nuclei. In terms of what tissues we have validated, many. We have validated colorectal, breast, lung, brain. So if you are interested in any specific tissue type, you can tell us, and then we can provide you additional information. So in terms of the solutions.
Sorry to interrupt, Tema. We have a question.
Yeah.
How do you obtain single cells?
How do we obtain single cells? So
yes.
Yeah, I will show the workflow in a couple of slides, if that's fine, and then we will see how we encapsulate the single cells. Oh, you mean from frozen tissue?
The question just says, how do you obtain single cells?
Okay. So I will go back to that when I show the workflow, and it will make more sense.
Okay, great.
Yep, thank you. So in terms of the solution, so Mission Bio offers the whole solution end-to-end, the instrument, the assays, and also the software for analysis. Our instrument is very, very small, very easy to use, and it works with disposable microfluidics cartridges. So you use one cartridge per sample. In terms of the reagents and assays, as I said, they can be catalog. I'll show you some examples, or also custom assays. And we provide you all the reagents you require from to prepare your cells suspension all the way to your sequencing ready library. In terms of the analysis, we have very friendly analysis solutions, and we train all our customers to perform the analysis on pipeline. We have 3 different pieces of software, all of them included in the price of the reagents, so you don't need to pay additional to analyze your data. We have a pipeline, and we have also very friendly software to generate these nice plots, as you can see here on the screen. In terms of how well it integrates to an existing NGS workflow, as you can see here, it integrates very seamlessly, because customers will prepare the cell suspension in the lab, then use the Tapestri to perform the encapsulation. And I'll show you in the next slides what I mean with that. Once you have your sequencing ready libraries, you will sequence them either in-house, or some people will send them to a core facility. And once you get the raw data, you use our software to analyze it. So it integrates very nicely in an existing NGS workflow. So in terms of how long does the assay take from the cell preparation all the way to library generation is 1.5 days. So you could essentially start on Monday with your cells and have your libraries ready to sequence on Tuesday, which is great. 1.5 days for DNA only, slightly longer for DNA plus protein, but it does not make a huge difference between DNA and DNA plus protein. So back to your questions on how you achieve single cell resolution. So this is what the workflow of the Tapestri platform looks like. So this is a two-step workflow. So customers will start either with cells in suspension or isolated nuclei. If they want to work with isolated nuclei, they need to, of course, isolate that. So get to the nuclei stage. We have universal nuclei isolation protocol that we have on our website and we can share to help people achieve single nuclei from a frozen tissue piece, from a frozen tissue slide. So in terms of single,
you answered the question. We got clarification. So the question is, how do you obtain single cells specifically from a frozen sample or a biopsy?
I see. Yeah. So we have a universal nuclei isolation protocol and outline step by step how you get to the single nuclei from a frozen tissue. You start by mincing the tissue, then we have some collagenase and disbase enzymes to release these single nuclei. And we also need to clean the solution and probably pass them through a strainer if we see there is debris or the nuclei are not in a monodispersed solution. So for single nuclei, it is important, and also for the single cell, right? As we work with microfluidics, the solution has to be monodispersed. Otherwise, we risk clogging the system. So yeah, that's, if you are more interested in that, we can provide the nuclei isolation protocol, which is publicly available on our website. So in terms of what happens within the cartridge and the instrument. So you get your cells in suspension or your single nuclei. And as I said, you work with disposable microfluidics cartridges. So this is what a representation of a cartridge. So what you do first, the first step is the encapsulation. So you get single cells here encapsulated with a protease solution in oil droplets, 40 micron oil droplets. This process only takes 5 minutes, and we follow a 5% Poisson distribution, which means that we generate millions of droplets and only 5% contain a cell. That is to avoid two cells being encapsulated together. So then we reduce the number of tablets. After we have our cells in suspension, we proceed to lysis. So as we encapsulate the cells with a protease, we need to lyse whatever sequesters the genomic DNA. So we proceed to lysis, and now we have an individual cell lysate for each single cell on your sample. After you have done the lysis, you introduce that again in a different part of the cartridge, where you combine it with a unique barcoding bits that have a unique barcode, and also all the necessary reagents that you require to do downstream amplification. So that is called barcoding, where you now combine a single cell lysate with a unique barcoding bit, so a bit with a unique barcode, and all the primers specific to your assay, because it is a targeted approach. So at the end of the day one, you generate like nano PCR reactors, where you have the genomic DNA of a single cell with a unique barcode and all the primers required to perform downstream amplification. So day one, it's around 4 hours in total, but hands-on time is less than an hour, really. The rest is all done by the instrument and thermocyclers. So for the DNA plus protein, and I will come back when I show you multi-omics data, we need to stain your cell suspension before you proceed to the encapsulation with something called antibody oligoconjugates. So the antibodies will be conjugated to an oligo tag, which is DNA. And then it gets, so the workflow is exactly the same. You see here, it just gets encapsulated. But at this point, when you do the lysis, when you do lysis, sorry, you don't get only the DNA, but also the tag for whatever antibodies your cell had. And then you continue downstream and generate two libraries, the DNA library and the protein library for a single cell. So it is true single cell. We are not talking about the same sample, but about the very, very same cell. So let's just look at the panels that we have available, the targeted single cell DNA panels. So just as an example, we have in hematology, AML, acute myeloid leukemia, we have a myeloid and also a CLL, chronic lymphocytic leukemia. These are our catalog panels, which are wet lab validated. For solid tumors, we have tumor hotspot panel and also the universal nuclei isolation protocol that I just mentioned before. As we have very, very good resolution at a single cell level, we also explore the gene editing world, and we have had very exciting and good results. So if you are interested in CRISPR-Cas or mouse and human models, we can also offer solutions for that, for gene editing. But for the sake of time, I'm only focusing in the single cell DNA targeted panels today. These are additional panels that we also provide. So if you recognize any of these as your disease or indication of interest, just so you know that we also have panels to help you, which have some content that you can explore. And if your area of interest is not here, if your indication of interest is not listed here, as I said, we can create a custom panel for you. How do we create a custom panel? So we have Tapestri Designer. This is an exceptional tool that we have online. It's free to register. You just go to tapestrydesigner.com and create an account with your email address. And then there you can explore the catalog panels and look at the content of these panels here, what mutations and what SNPs are present. And if you're not, if you don't find the indication of your interest, we can also create a custom panel for you. That is also recommended for people working with CRISPR-Cas and also working with mouse and working with a CNV, if they want to do like a whole chromosome, let's say, or many chromosomes approach. So I don't know, this is the end of the platform overview. I don't know if at this point anybody has any questions before I move on to real data.
I think we're good.
Perfect. So then let's just move on to the first customer case. So this is the resistance to targeted therapy. As I said, I'm very excited to be presenting this because it is the first time I show it. It's been recently published in Blood Advances. So this is a paper from our customers called Agios. This is a pharmaceutical company in America that developed a drug called ivosidenib, which is an inhibitor for IDH1. Ivosidenib has been approved to treat refractory AML, and IDH1 mutations occur in 6 to 10% of patients with AML. So the project interest, so this is a follow-up study in 174 patients, and they wanted to assess molecular mechanisms of the ivosidenib resistance. The key questions of this study were, what is the clonal architecture of underlying resistance? And also, how do the dynamics change from baseline to relapse? This is a very exciting landscape. This is bulk sequencing data right now. It is not yet single cell, but it is a very interesting landscape to look at because these are the 174 patients organized based on prognosis or response to the therapy. So on the very left, they are the ones that achieved clinical remission. And on the right are the ones with the worst outcomes, so the non-responders. Also, the mutations are organized from top to bottom, bucketed in different pathways where they take part in. So as you can see, all the patients are IDH1 mutant. That's because they selected a cohort of patients that are IDH1 positive, of course, because they are studying the resistance to ivosidenib. So what you can see here is the non-responders are actually the larger buckets with the majority of the cell with a larger number of genes being affected. You can see here the IDH1 mutants and also the co-current mutations with other genes below from many different pathways. And interestingly, same as with the Kathy Smith and Alexander Pearl's data, they also see activations of the RTK pathway in the non-responders bucket. So it is a very nice figure to see 174 patients at once and how the non-responders happen to be the more complex. So you expect that to be more clonal and have more heterogeneity. And that's why that probably is the reason why they don't respond to therapy. So as I said, this is bulk sequencing data, but now after they have all the information about the 174 patients, they selected some to do single cell analysis. So which patients they selected? They selected the patients that have IDH1 and IDH2 together. Why? Because they also have a drug against IDH2. So they just took a subset of patients that had IDH2 mutations, and they wanted to see the clonal architecture of these patients. So they analyze, of course, different time points, 3 time points per patient. So as they do bulk sequencing, they know which ones have IDH2 mutations. So I will show you now 3 different scenarios that they saw of these patients with IDH1 and IDH2. So first scenario, 6 out of 9 patients have IDH1 and IDH2 co-current mutations. As you can see here, these are time point 1, time point 2, time point 3. At time point 1, this patient had IDH1, NPM1, and RAS, IDH1, NPM1, FLT3 tyrosine kinase domain. And then this clone is the one acquiring IDH2 at the time of relapse. So actually it is a co-occurrence of IDH2 and IDH1 at this time point. This happens, so IDH1 and IDH2 co-occurrent mutations happened in 6 out of 9 patients. Another interesting scenario is something called isoform switching. Here we see IDH1 is present at time point 1, but then disappears or almost disappears at time point 2, where IDH2 arises. So here is IDH2 is happening, is just being acquired while IDH2 is disappearing. This is another mechanisms of resistance to this drug, and this happens in 2 out of 9 patients. So if we look at the third scenario, we can see IDH2 at this point was actually detected at baseline with single cell, but not with bulk. So with bulk, that is what I mentioned in the first slide. The IDH2 was present in such a small percentage that bulk couldn't detect it at this point. But with single cell, we see that actually IDH2 was present at baseline. And it is a completely different clone. You can see here, IDH2 and IDH1 are different clones. What they also found at this point is that at the relapse, the NRAS and ASXL1 double mutants with IDH1 happens to be the predominant clone in terms of resistance rather than IDH2. So what this is showing is first, this would be impossible to detect with if not using single cell DNA, because you don't have the information of the clonality. You could infer based on allele frequencies, but you don't know whether they happen in the same or different population. And second, the resistance to this drug is very complex. I mean, it is polyclonal, and these patients might benefit of a dual therapy perhaps. So that's why they were so interested in looking at these patients this way, because as I said, they also have a drug against IDH2. So now, as you know the clonality and how these patients or these clones will behave, you can, predict whether they will be candidates as well for a dual therapy with ivosidenib and whatever drugs for IDH2. I don't know if there is any questions at this point about this study.
No, we're good.
Perfect. So that was in like DNA only therapy resistance. And now I will move on to multiomics. So as you might know, Multimodal single-cell sequencing was named by Nature Methods as the method of the year in 2020. So we are so excited to be part of this exciting application, because we have now SNV, CNV, and protein. So starting with the CNV approach, just to give you a quick overview. So the single cell CNV solution is targeted. That means that you can direct the amplicons to the regions you are truly interested in. So you get the maximum coverage with the maximum resolution at the minimum sequencing costs. So if you have at least three amplicons per target is recommended for single pol mutations or like a gene. And you could also have, if you are interested in whole chromosome or whole arm, you can also have 5 to 12 amplicons per region, and then just focus on these chromosomal gains or losses that you are used to see in your specific cohort. So we have a very, very nice resolution, so 20 kV, and also a very good coverage, 40 to 80x, which if you wanted to achieve 40 to 80x for whole genome, that would be crazy if you wanted to achieve that with sequencing for a whole genome approach. So I mean, here we have, like, you can look at, as I said, single loci, single gene, single exon, single chromosome, or even multiple chromosomes, so chromosomes arms, thanks to the, the flexibility of our custom panels. Unlike with whole genome sequencing, that you can look at bigger events, single cell whole genome sequencing, bigger events, but you'd get less resolutions. So on top of all of these, remember that we also provide genotype information. So not because you get a CNV solution, you won't get the genotype. You will get all the genotype information that I showed you for the previous slide, together with whatever chromosomal rearrangements you are interested to analyze. So let me just show you an example of real data of the copy number assay. So this is a collaboration with Charlie Swanton's lab at the Crick Institute, and this is renal carcinoma. So I wanted to show you some nuclei data as well, extracted nuclei data. So this is renal carcinoma extracted nuclei, and they assessed copy number changes in chromosome 3, 9, and 14, because these are the chromosomes that are often lost in renal carcinoma. So as you can see here, this is the cells represented as a heat map. And it is important to highlight that we don't need 2 samples in order to achieve copy number. So from the very sample, remember, we get genotype information, right? So we determine what population is the control population or wild type population within your sample, and we can normalize against this normal population. That's what we see here. So for this renal carcinoma sample, we have 4,500 cells, and 1,500 are normal, with no copy number changes. So if we normalize against the normal population, then we start seeing the copy number changes for the rest of the population. So we see here clearly there are losses in chromosome 3, 9, and 14 that are also represented by gene in this line plot on the right. So again, this is not only what we provide, because we provide you the genotype in addition to the copy number, and we will see it when I show you now the multiomics analysis.
And a quick question. You mentioned the need for a normal population. What if your sample doesn't have a normal population? Could we still use the CMV assay?
Yeah, that's a question that we get very often. So we have tested internally as little as 5% of a normal population will be enough for you to call copy number. If you are working with a high tumor burden sample or a pre-sorted population, so you expect 100% to be tumor, a solution that you could do would be a spike in. I know not everybody want to use a spike in solution, but if it is a 100% tumor, which is not often the case, that is 100%, that would be something that you could try, and spiking of a normal cell line or normal, population, just to do the normalization.
Great, thank you.
Good. Perfect. So if there are no questions about this, no more questions about the CNV only, let's move on to CNV, SNV, and protein. So very excited to talk about this. We are the first company providing genotype to phenotype capabilities. And we have a partnership with BioLegend for our customers to purchase the antibodies for the multiomics assay. So the output of the DNA plus protein workflow is between 2,000 and 10,000 cells, and we could do up to 1,000 applicants. So how many antibodies you can do at once? You can multiplex up to 40 or plus antibodies for cell surface protein expression. And as I said, in terms of the workflow, it adds a little bit of time at the beginning for staining, around 1 hour, because of course you have to stain your cells prior to the encapsulation with these antibodies oligoconjugates. Then you perform the assay very similarly to what I described for DNA, but another difference is that you generate 2 libraries per sample. So you have your DNA library and your protein library that afterwards bioinformatically you will combine, so you know which protein and which mutations are coming from the exact same cell. So that is what I will show you here. Just to finish off with the presentation, this is a collaboration with Dr. Chris Horrigan from the NIH. So this is a real AML sample, and they generated a custom panel, a custom DNA panel for 45 applicants with the following antibodies, CD3, CD11B, 9, 34, 38, and 90. So first of all, just to tell you, I mean, as we get 3 different analytes, it is possible to cluster your data based on any of the analytes that we generate. So protein, SNV, or CNV. Here you can see TSNE plots of the data or the cells clustered by SNV, protein, and CNV. So in this case, you can see that the protein is giving you 4 different populations. So you see a cancer population, and then the T cells and the B cells based on cell membrane markers. If you cluster of SNV only, you get 2 populations. You get the cancer population and the wild type population. But of course, you don't have information about what population that is. If you try to cluster on CNV, at this point, there was not enough data to separate the populations, despite of having some copy number changes. You can see the blue and green are kind of grouped in this area, but it is not enough as to separate them. So based on this data, we decided to cluster based on protein. So this is what the results look like. And don't worry, I know it is busy, but I will break it into different sections. So here, let's just look at the protein information first. So based on the proteins, as you see in this TSNE, we have 4 different populations. We have the main cancer population with CD11B negative, 34, 38, 90 positive, which is almost 4,800 cells. Then we have a cancer subpopulation, which is CD11B positive, CD3, 180 cells. And then we have the healthy cells, which are the T cells, CD3, and B cells, CD19. So this is based on the protein. You see here the color code for the protein expression. Yellow means high, blue means low. We also have copy number, right? So the copy number information, yellow means a gain, and then dark blue is a loss. And we also have the genotype, SNV, and indels. So dark blue is wild type, light blue is heterozygous, turquoise is homozygous. So let's just look at this SNV first. So the genotype information first. What is the mutation that is different in the cancer cells and the healthy cells? So we see here, if we looked at the ASXL1 gene, there is this specific mutation, this specific locus that goes from wild type, dark blue, to heterozygous, light blue. So that is a pathogenic ASXL1 mutation present in both of the cancer populations and not in the healthy cells. So that's that will be just generating this cancer cluster that will be different to the healthy cluster. That in terms of SNV, yes, SNV. Now CNV. Look at this stretch here. So you see that the healthy cells are copy number 2, and now we see a copy number 1. So there is a copy number loss. The interesting thing about this is as I said, we don't stop here. We provide you the genotype associated to these copy number changes. So if you go to copy number 2 to copy number 1, you would expect the genotype to change. And that's what we see here. This is exactly the same region of the chromosome 7. So when you go from copy number 2 to copy number 1, This mutation over here goes from heterozygous to homozygous. So there we have lost the wild type allele. Likewise, here, this mutation goes from heterozygous to wild type. So here, combined to the copy number loss, we have also lost the mutant allele. So it is very exciting to see the genotype associated to the copy number in real time, something that you couldn't see with a copy number only assay. And that also is supporting information that this copy number is real because it has a genotype associated to it. So additional to that copy number, we also see another copy number lost in chromosome 4. So that's, I just want to highlight the amount of information that you get, how exciting this is, that you get it in 1.5 days from a single sample with a single assay. So with a total of 4 or 5 hours hands-on, in a couple of days, you get this array of information of protein, cell population, copy number, and genotype from a single sample. So I'm going to show you before I finish, because I imagine that some of you might be working with flow cytometry, and this is a question that I get all the time when I present this data, how well this correlates with flow cytometry. So I'll show you this slide. It is from an internal experiment of cell lines where we just pretty much mixed 5 cell lines and proceeded to do the Mission Bio assay at the same time as we do flow cytometry assay. And we measure the protein expression for different markers. So here you have the 5 different cell lines, and here you have the 6 different markers. And this is Mission Bio, and this is flow cytometry. So we have just highlighted one cell line and one marker to make it easier, so you can see the correlation of the protein expression of the multi-omics assay with the Tapestri platform versus flow cytometry, if that is what you are more used to see. So with all that, let me just summarize that the Tapestri, it is the only platform that provides you DNA plus protein from the very same cell. And when I say DNA, I talk about SNVs, indels, and also copy number. It only adds a simple cell staining process upfront, which is an hour. To the Tapestri platforms, you can still fit it in 2 days easily. And it is being validated on cell lines and blood cancer samples. And of course, we provide you information from genotypes to phenotypes. So just to finish off, I'll show you the number of publications that we have from 2018. So this is 2018 to 2019. I'm going to just show you quickly. We can share the slides if you want to. These are publications in 2020 and more publications to come. So as you can see, we're very excited that our customers are starting to publish a lot of studies, and many of them already have DNA plus protein. So with that, I'd like to thank you for your attention and see if you have any questions.
Emma, that was wonderful. Thank you. So we've gotten a lot of great questions coming in, and I think we'll go backwards. So from the data that you showed with chromosome 4 loss, is the chromosome 4 copy loss also reflected on the SNV and indels?
For the Chris Horrigan, for this one?
Correct, yes.
Yeah. So in this plot, we don't have information on the chromosome 4, I believe, because the custom DNA panel is only 45 amplicons. So probably there was no genotypes changes or no common variants detected in that chromosome 4 stretch. That's why I don't show it. If the panel was bigger, we probably have seen changes in genotype for that region. I mean, we are using GATK to call genotypes, so probably there was no variants or changes in that specific area of the chromosome 4.
As a follow-up to that, then how many amplicons would you recommend to cover a certain region?
Yeah, it depends. So if you are interested in a specific somatic mutation, as an example, HER2 superamplification in breast cancer, you could get away with 3, 1 for your mutation and 2 in the flanking regions. But as usual, the more the better. So if you want to study a larger region, like a chromosome arm, 5 to 12 will be great. Although if you are studying presence absence, you could also, you could be okay with 5 or 6. It also goes down to cost, right? So the more applicants you have, the more information, but it is not necessary to have 200 applicants to assess presence absence. Probably, 5 or 10 will be enough.
Wow. So a follow-up to that, how do you assess the CMVs? Isn't the workflow PCR based?
Yes, it is PCR based, but as I said, so what we do, because we have information of the genotype, we determine what is the control population, and your control is part of the same sample. So we just normalize the applicant signal against the normal population. So we start seeing the copy number changes.
Great. And then has someone tracked any transcription factors along with SMVs?
I don't recall that by heart myself of any of my customers, but we can definitely check internally if that has been done and get back to the person who asked the question.
Great. Do you plan to add epigenetic information to the list of readouts?
Yeah, I mean, we would love to. We would love to add everything. At this point, we are all focused on DNA plus protein, probably intracellular proteins in the future. Epigenetics, not that I know of in an immediate future, but definitely we will look at it, same as with other analytes as well.
Yes, and I think what's exciting for us by adding protein, it's similar to RNA expression in the sense that now we can identify cell type, and that's been huge for these samples. So for instance, in this plot, even though they have the same genotype and they're indicating that they're cancerous via their genotype, the protein is identifying two different populations. I just find incredibly powerful.
Yeah, it is exciting.
All right. Well, I don't think we have any more questions. They were wonderful questions. And Hema, thank you so much for taking us, giving us an overview of the Tapestri platform.
Thank you so much. Have a good day.
Good day, everyone.
Bye-bye.